Well, when it comes to cell signaling, we first talk about our own signaling. When we want to communicate a message to another person, we have lots of different methods. We can write a letter, we can send a text, an email, we can even send smoke signals. Cells, similarly, have a lot of different ways they can send signals between each other, whether those are hormones or autocrine signals, different chemicals, or a nerve signal. But pretty much they all come down to a chemical signal of some sort. And that chemical signal is received by a receptor. One important model for cell signaling, particularly receiving the signal, is the G protein coupled receptor. And that's just because this receptor is coupled to a G protein. Simple as that. Aren't they a cute couple? I know, totes adorbs. Let's take a look. And there's a lot of different kinds of receptors out there. A lot of times they're drawn like this because they're these chains of amino acids that snake back and forth through the membrane. That's kind of what holds them in place. All these hydrophobic amino acids in here like to stay in the membrane. But this is the site where the signal molecule sits. Every cell in our body is coated with thousands of receptors, thousands of these proteins, and often hundreds of types of them on the surface of the cell. So it's just like thousands of ears all over the place on a cell, listening for these chemical signals. And each one is tuned to just one specific chemical. And when this chemical binds here, it's kind of like the ringing of a doorbell at that entrance to the cell, saying, hey cell, you need to do something. And so we're going to go through a model of how that happens. And this specific set of G protein coupled receptors is involved in some types of taste, smell, and a lot of the nerve signals that go out, especially those in the fight or flight system of our nervous system, use this signaling pathway. And a lot of the medicines, at least the modern medicines that are prescribed, also target these G protein coupled receptors. Well, some mechanisms of receptors are really simple. They just open up an ion channel, ions flood into the cell and change the membrane potential, causing a response. The G protein coupled receptor is quite a bit more complicated. There's a chain reaction of chemical events. And so there's a couple things that I want you to look at in this animation I've prepared about how this works. And this is just the start of it. So we have this signal molecule. Is the thing bringing the message to this cell? This represents the cell membrane. And it's saying, hey, you cell, I want you to take this action. But the surface of the cell can't do anything. We need to get that signal down in here. So that process of getting the signal into the cell is signal transduction. And that's the one that takes place in this chain reaction of chemical events. All right, so one reaction kicks off another reaction, kicks off another reaction. So expect to see that one. The other thing that you'll see is signal amplification. The signal gets bigger and bigger and bigger as it comes in. We start off with one signal molecule, but sooner or later inside we're going to have thousands of signal molecules amplifying that signal, just making it bigger so that the cell can have a bigger response. And a lot of that is because some of these chemical reactions are helped out by enzymes. You know what enzymes do. They crank out product after product after product after product after product. So when it goes from step 1 to step 2, that enzyme might magnify the signal 100 times. So watch for those things in this animation. It's a little bit basic, I suppose, but it gets the point across. I'll go along at a fairly quick pace. Normally I'd take it really slow and methodically, but since this is a video, I'm going to assume that you're going to back this up and watch it again to really let this sink in. This is not one of those things that you watch once and say, got it, great, I can just narrate the whole thing from memory right now. That may not necessarily be the goal, But I do want you to understand especially these two processes. The other thing I want you to watch for as we go along is old tools used in new ways. And here's my analogy for that. I was wandering around one day carrying a hammer, and I hammered it into the ground and found out this is a really excellent weeder. So this old hammer of mine, even though it's not its purpose, is one of my favorite weeding tools in the lawn. So there you go. Old tool repurposed. You're going to see a few tools, enzymes and especially ATP, that you're familiar with, but they're used in a new and somewhat unfamiliar context and in somewhat new ways. And so there's actually not a whole lot of new stuff to learn, but you're going to need to be flexible in your thinking and sort of adapt and broaden your horizons as to how those molecules are used. Here we go. Let's start off with some of the cast of characters. An oldie but a goodie. ATP, adenosine triphosphate with its 1, 2, 3 phosphates. Break off a phosphate and you get adenosine diphosphate, just 2 of them. Break off one more phosphate and you get adenosine monophosphate. But then that does something weird with this phosphate. It creates this loop, this circle. And so this one is cyclic adenosine monophosphate. So that's what the C stands for. Some people just call it CAMP. There's the cast of characters. Let's move on to the process. So here's our receptor, G protein standing by, and the signal molecule. And the goal of this entire process is this signal molecule is being sent so that it will activate this enzyme, which is currently inactive. Its job is to break down this polymer. Let's just say it's glycogen and it's supposed to break it up into glucose. All right, so there's the setup. Here's how that happens. The signal molecule binds. G proteins are activated and they actually remain along the membrane and activate this other enzyme, adenylate cyclase. And there's a bunch of them, so as long as that receptor stays bound to the signal molecule, these G proteins keep getting activated. And now these enzymes have been turned on. They're ready to go. They were inactive and now they are active. Their job is to take our old friend ATP and convert it to cyclic AMP. So there it binds, now it's cyclic AMP, and that product is released. And this can happen again and again and again. Binds, converts it, and that's released, and that could keep going. Here's that amplification. So these are new signal molecules. We had 1 original, and now we have 6 in here. In a real cell, there might be dozens or even hundreds of them. All right, what is that cyclic AMP signal? It signals another enzyme, protein kinase A. You can tell it's an enzyme because its name ends in ASE. So there it is. Its job, and there's bunches of them, I haven't drawn them in with these other cyclic AMPs, but they would be activating those as well, is to bind to the enzyme and then take another ATP and bind to it and add the phosphate to the enzyme, which now makes it active. So here's its active form. You can see we change that active site, and now it can go off and do its job and start munching away at this polymer, breaking it down into a bunch of monomers that the cell can use. And the other amplification step that I didn't show here is that each of these protein kinase A's, since they are enzymes, can react dozens or hundreds of times themselves. Frankly, it's just too big of an animation task to do that. But this protein kinase A might phosphorylate, let's say, 100 enzymes. And this one might activate 100 more. And this one, 100 more. We only activated 1. So you can see how big this signal could really grow within the cell. And now we have accomplished our mission. This signal molecule was sent to activate an enzyme, and it has. Quick recap. We started with the receptor, a signal molecule bound to it, which activated a G protein, which activated an enzyme, adenylate cyclase, which produced cyclic AMP, which activated another enzyme, protein kinase A, which bound to an enzyme and phosphorylated it using ATP so that it could break down a polymer. Well, that is a lot to take in. If you got to back up and watch it a few more times, I think that's probably a good idea. Hopefully you're catching these major themes as you go along. Now, this is an important process. It's part of how our bodies coordinate all of our different cells. We're 30 trillion cells acting as one unit. And it's this sort of signaling to each other, just like a big company or a team, good communication is key to that function. And this is one of those things. So this is probably a good preview of quite a bit of what's to come in anatomy and physiology for those of you moving on to that class. You've seen a lot of different cellular and molecular tools that you're going to see again when you get to anatomy and physiology. You're going to see some new ways that they're used and the combination of different tools into more complex actions. And I hope that sort of complexity, even though it may sound challenging, appeals to you in some way. It is fascinating. Well, that's it for this time. Until next time, keep appreciating life, even if it's a little confusing.